All
There.
right. Welcome back, everybody, to the third talk of the afternoon session. It is my absolute pleasure to introduce Dr. Becket Ebitz, who recently joined the University of Montreal. This was a long hiring process, which I was happy to be involved with sort of peripherally, but I'm super glad he's with us. So Becket is a cognitive and computational neuroscientist who's recently joined the Department of Neuroscience at University de Montréal. Previously, he worked as a senior researcher at the University of Minnesota and has a postdoctoral fellow. He was a postdoctoral fellow at Stanford, where he studied reward learning and implementation by neural circuit dynamics within the brain. He also held a CV Star Fellow position at Princeton University, working on learning and strategic decision making. His main research interests are learning, decision making, and executive control. And today, Becket is going to talk to us about the neuroscience of exploration. So, take it away, Becket.
Okay. All right. So I'm thrilled to be here and to explain a little bit about what I think of when I hear the word exploration. And I think people use this word to mean a lot of different things, but in general, what it has in common is that it's the solution to this kind of question. How do we navigate uncertain task spaces? So task spaces where we don't know what the most rewarding thing to do is. And task spaces can be uncertain for a number of different reasons. They can be uncertain because they're new, because you're just dropped down into some world and you have to figure out how to navigate to some rewarding location in the world. But they can also be uncertain because they're changing, because over time, either because the world is dynamic or because your needs change over time, the best thing to do is shifting. And I think in general, there are algorithms that can actually simultaneously solve both of these problems. That can solve the problem of exploring new spaces and changing spaces. But they require that exploration not be occurring at the level of like individual muscle activations. That's just sort of too high dimensional for these algorithms to work really well. Instead, if we sort of parse the world intelligently and plan or explore in directions of travel, say something that's quite a bit lower dimensional, we can do quite well in these spaces. So I'm going to break these two things apart today and tell you two different stories. The first one that I'm going to tell you is about how mice navigate new environments. And the second one that I'm going to tell you is about how monkeys navigate changing environments. Okay, so let's start with the first story, the story about mice. So in general, the world is quite a bit more complicated than this like two-dimensional landscape with like, an easy to learn gradient that I've illustrated here. Real environments have quite a bit more dimensions than two. And as a result, there are sort of a lot of work on exploration has focused on this problem of exploring intelligently in really complex high-dimensional environments, environments where rewards might be sparse or something like that. And I think a lot of that work sort of is based around the same idea that we would use information maximizing strategies, what's known as directed exploration in the cognitive science literature, to sort of figure out what direction you want to travel in. So that's not what I'm going to talk about today, but if it's something you're interested in, I would recommend you look at Bob Wilson's work or Sam Gershman. And there are a couple of good reviews, one by Sam Gershman and one that Bob and I wrote along with Vinny Costa and Liz Bonowitz that recently came out that I would recommend you look at. Instead, what I'm going to talk about today is how we can learn new spaces by sort of reducing the dimensionality of the space, by constraining exploration. And so the first story that I'm going to tell you is work that I did with Nicola Grissom and Kathy Chen at the University of Minnesota. And what they did was get mice to do what seemed initially a very simple task. So the mice just have to choose between two giant visual stimuli, one of which is associated with an 80% chance of reward and one of which is associated with a 20% chance of reward. So it seems, like this is something you would be able to do. You just pick the most rewarding image and then you sort of learn it very quickly. And so at first blush, this seems like, an incredibly simple task. It basically has one dimension. You either pick the spider or pick the scissors. But mice evolved in their own sort of world And they might consider other dimensions that we're not considering when we design the task, like the location that the images are in, or even more complex transformations, like the interaction between images and locations, or even things that change over time. So the interaction between image locations and whether they were rewarded on the last trial, or even like features of their movement on the last trial. There's really sort of no upper bound on how complex you can make this task if you wanted to make it really complex. So the first thing that Kathy and Nicola did was look at how quickly mice learn in this environment. And there are a couple of things that I want you to note from this graph. The first is that it actually took them a really long time to learn this task. So each bin here is about 150 trials, which means it took them about 2,000 trials to acquire this really seemingly simple association. And I should say that mice are not visual animals, so something that looks super obvious to us is more of a fine visual discrimination for the mouse. So maybe it's not that surprising that it took them that long to acquire it. But the other thing that they noticed was that the female mice actually acquired this association a bit faster than the males. And that's the part that I'm going to sort of answer today. Why did the female mice learn faster than the males? So the first thing that we did to try to make sense out of how the mice were learning this task was to sort of think about what kinds of strategies you could use over time as you see these options presented on the screen. So, on the left, there's a scissors, on the right, there's a spider, and then over time, you have to sort of apply some strategy. The best thing to do is just to pick the most rewarding image, pick the spider here. But you could also use some strategy that just repeats in one spatial dimension and just takes whatever rewards you get for doing that. You can also imagine much more complicated strategies, like reward-dependent strategies, like win, stay, lose, shift strategies, which again can happen either in the image domain or in the spatial domain. And what we found when we looked at the extent to which mice use these different strategies over time was that they actually process through applying different strategies. Ultimately, they sort of end up doing the best thing, which is repeating the correct image with some bias based on the reward of the last trial. But the other thing that we noticed was that they generally start with this initial spatial bias where they just choose one side over and over again. The other thing that we noticed was that this pattern was really driven by the female animals. The males didn't, as a group, have as much of this early spatial bias. And we think that this early spatial bias was actually jump-starting the learning in the females because it constrained the dimensionality of the task. So they just got to observe the associations with each image and hold many other things constant, like the actions that they were doing on each trial. And we think that that really accelerated it. The males, on the other hand, were not more random. So it wasn't just that they were bumbling about trying to figure out what was going on, but instead they were sequencing through like really complicated hypotheses, hypotheses that were actually difficult to explain in like natural language. So we figured this out by sort of expressing their choice pattern as some probability distribution over all things that they could be doing. And so I had said that they are not more random. They're not just choosing sort of uniformly over the space of possibilities, but their choices were also more organized with respect to the last outcome and the last choice than the females. So they were using these much more sort of precise strategies than the females. The other thing we noticed, if we express these probability distributions now as probability vectors on the simplex of strategies, was that we can observe how they're evolving these sort of agnostic strategies over time as they approach the best thing to do, this little star here. When we compared the males and females in this sort of space, what we discovered was that females were taking this sort of stereotype procession every time they learned a new image pair, where they initially held the dimensionality of the task, constrained, and then eventually learned the right thing to do. Whereas males were taking these giant leaps from moment to moment and doing a different thing every time that they went through the task, suggesting that their strategies were a lot more idiosyncratic and high dimensional than the females. So just to summarize this part of the task, we found that learning was surprisingly structured in mice. They didn't just acquire the ultimate association that we wanted them to, but instead sort of bootstrapped it through successive strategies over time. We found that males and females both made completely incorrect assumptions about the dimensionality of the task, but that the female approach happened to be better suited for the task that we gave them. We think that different approaches, maybe the male approach might be better suited for environments where rewards are more sparse, or when rewards are some function of the interaction of multiple dimensions of the stimuli. But this kind of inter-individual variability could be a powerful way to sort of ensure that the species as a whole can find good things to do in the world. So I would argue that this sort of circumstance where you're plunked down in a bewildering new environment and you have to figure it out is not something that we necessarily encounter every day. But I think that this second problem, navigating changing spaces, is really the kind of exploration that we do almost all of the time. And I think in this context, what I mean when I say exploration is basically any pattern of behavior, whatever it may look like, that facilitates transitions between sort of temporally extended policies that we might be adhering to. So to unpack that a little bit, I have to tell you a little bit about how I think we make decisions about what to do. There's definitely compelling evidence at this point that we decide what to do based on the value of doing so. So if we're comparing different policies and we think policy 1 is more valuable than policy 2, then we'll do policy 1. But the other thing that we notice if we look at animal behavior and human behavior is that people tend to be sort of a lot stickier than you would necessarily expect from a simple value account. And if you want to make like a simple reinforcement learning model, model-free temporal difference learning model, fit behavior in monkeys and humans, you have to add these addenda that basically make it more sticky to account for this observation. So you have to make the model hysteretic, or you have to make it learn more from wins than losses, right? And these things all serve this purpose of sort of making behavior more sticky to account for this fact that we decide on our policies not just based on the behavior, but based on sort of the energetic costs of switching back and forth between policies. So in monkeys, if we want to study exploration, we have to start with some kind of a task. And so I'm a monkey electrophysiologist, so a lot like what Nicole Rust was talking about earlier today. What I do is ask monkeys to make decisions by moving their eyes in various directions in space while I give them rewards for doing things. So in this task, we just have monkeys choosing between two different options, and we either give them a reward or don't give them a reward based on the option that they chose and the probability of reward associated with that option. So here we just have two targets whose reward values, the probability that you'll get rewarded for choosing each of them, just drifts slowly and independently over time. And what this does is encourage the monkey to both exploit an option when it's good, right? When its value is high, it's rewarding, the monkey wants to sample it as much as possible, but also to occasionally stop exploiting some option in order to explore the alternatives in the environment. And what we did, which is sort of reviewed in this paper here from 2018, is really look at the dynamics of the choice sequences of behavior in this task. So looking at the duration of different runs of different choices, and asking if that was structured in some way. And the structure that we found was that there are sort of three different latent states going on in this task. There's one state where they're sort of repeatedly choosing option 1, which we call exploit target 1 or oit target 1, and one where they're repeatedly choosing target 2, which we call exploit target 2. But there's also this third state, which in this particular task happens to be random selection between the set of available options. So we call that the exploratory state. And what this means, if we sort of think about this dynamical landscape that I drew before, is that exploration, in this space anyway, is this little divot in between exploiting one thing and exploiting another thing. And we know from sort of protein folding work that having this extra little divot between two different states that we want to be in can actually facilitate transitions back and forth between these different policies. So I should say also that it doesn't have to be random. So it happened to be random in this particular task. But in more complex tasks where the choice is some function of the color or the shape of the option, where it's some complex rule, what we find is that monkeys actually explore using this very directed sequential hypothesis testing strategy. And it turns out that making the same sort of emissions model, this same sort of uniform across the set of available options, also allows you to effectively recover exploration in this context. And that's just because it's the maximum entropy distribution for the choices. So this general approach works well, regardless of whether you're looking at random exploration where you're sampling randomly or directed exploration where you're doing some sort of information maximizing, complex strategy. So one thing I really like about this approach, about finding these latent states, finding this kind of structure underlying behavior, is that we can basically describe behavior, the transitions between different states over time, in terms of like a set of equations that map behavior at one time point to behavior at another time point, right? We have a transmission matrix here. And then we can analyze that transmission matrix and make statements about how different things are changing the dynamics of behavior. So one thing that we've done here is in that rule-based decision-making task, where you're making decisions about colors or shapes or something like that, we can measure the dynamics of behavior where you have color rules and shape rules. And what we find is that getting a reward sort of deepens these basins that correspond to rules, right? It stabilizes rule states, whereas not getting a reward makes them sort of flatten out and makes it easier to transition into exploration. But then we can do something like give monkeys cocaine and figure out what that's doing to the landscape of behavior. And what we find is that cocaine really specifically stabilizes rule states, but doesn't do anything else to the behavior. We can also use this to make sense of individual differences. So with Kathy and Nicola at the University of Minnesota, we found that the male mice tend to explore about 15% more than female mice. And this seems to be driven by the fact that exploration is just a more stable state for the males. It's harder for males to get out of exploring than it is for females, at least in this particular sample. I think one cool thing about this view, about thinking about the dynamics of the latent states underlying behavior, is that it also makes a couple of predictions about when exploration should occur and how costly exploration should be. Specifically, it predicts that some of our transitions into exploration are driven by noise, right? They're made more likely when you haven't gotten a reward. It's easier to get into exploration, but the driving force is really this sort of diffusive noise process that's causing us to get into exploration. And the other thing is that exploration is sort of globally costly. At least in humans and monkeys, it actually doesn't appear to be that way in mice, but the idea is that exploration is this globally unstable state. You need to add energy into the system to get it to explore. And so we can actually look for these kinds of predictions in data, and we find some evidence that these things are true. So we've got this PLOS computational biology paper, which suggests that exploration is occurring tonically, both when it's useful and when it's not. And we do that by giving these long periods where nothing is changing, and the monkeys have overlearned the task. They know that nothing's changing. They still lapse. They still deviate from what they should be doing. But it turns out that that same underlying process that's causing these lapses, that's causing them to make mistakes when exploration has no value, is also driving their ability to adapt at change points, right? So there's some one-dimensional manifold that relates those two, some underlying exploratory process driving both of those behaviors. And what cocaine is actually doing in this task is pushing them down along this one-dimensional manifold. It's regulating that underlying exploratory process that's responsible for both of these things. We can also say that exploration is costly. So we have some nice sort of behavioral data that suggests that in order to start exploring, we have to give you a big burst of information, but you actually tend to just fall out of exploration. At least humans and monkeys do. Mice, you have to give them more information to get them to come out of exploration. But if we look in the brain, we can actually see that exploration is energetically costly in terms of its metabolic costs as well, in terms of the number of spikes that we expend making exploratory choices. And this is true across a bunch of brain regions that we've looked at: orbital frontal cortex, ventral striatum, and dorsal striatum. Exploratory decisions require more spikes. And they're also a less efficient use of activity. So the amount of information about choice that we have per spike is actually lower during exploratory choices. So I should say here we call these rule-free choices because it's a set of choices, the majority of which are exploratory, but not all of which are exploratory. So beyond allowing us to start thinking about the dynamics of behavior and thinking about behavior decision-making, sequential decision-making as some diffusive process in a dynamical landscape, The models also just give us labels. They give us the ability to say whether a given choice was likely to be due to an exploratory state or an exploitative one. And because I'm a primate electrophysiologist and I'm recording from neural activity in monkeys while they're making these decisions, that's really useful for me, because now I can compare the neural correlates of exploration and exploitation and say something, finally, now that we're more than halfway done with the talk, about how exploration is implemented in the brain. So I should say that in general, these labels that we're inferring from these hidden Markov models, these latent states explain a ton of variance in neural activity. So they explain more variance in neural activity than reward value, than whether it's a switch or state decision. And in this task that I've mentioned a few times, where we're looking at the difference between color rules and shape rules, different kinds of like higher order cognitive rules, we actually find that something like 80% of neurons across these 3 different brain regions just encodes the identity of the rule that we discover using these kinds of latent state models. So it's really a very powerful approach. But what I want to talk about in my remaining time is basically how these states are changing information processing within this part of the prefrontal cortex that's responsible for mapping incoming sensory information onto decisions about what to do. So this is a part of the brain called the frontal eye fields. And you don't need to know anything about the frontal eye fields except to know that neurons within the frontal eye fields predict the choice that you're going to make. So they have spatial receptive fields. They have locations in the world that they care about. And if you're going to choose an object that's in their receptive fields, you'll get a lot of activity in a frontal eye field neuron compared to if you're going to choose any other target in the visual field, anything that's not in the receptive field neuron. But it turns out that that's basically true during exploitation, which I would argue is what almost every laboratory task that we do in monkeys requires. We require animals to exploit the rewards that we've told them about in the world. But during exploration, we actually lose quite a bit of this choice predictive activity in the frontal eye fields. This is true when we average across the entire population of neurons as well. That was a sort of single example neuron that I was showing you before. And now this is the average over however many neurons I recorded. I'm sorry, this was a few years back now. But one of the things that we're doing here is actually recording simultaneously from a bunch of neurons at the same time. And that allows us to ask slightly more sophisticated questions. So here, if you imagine the activity of all of our simultaneously recorded neurons is some dimension, then what we're thinking about is neural activity at some point in a neuron dimensional state space, right? And that point corresponds to some representation of something. So here, what we're doing is finding the sort of low dimensional projections in neuron dimensional space that predict the choice the animal is going to make. So that happens to correspond to these vectors here. So these are ideal amplifying vectors along which neural activity would evolve over time if it became increasingly predictive of the choices the monkey was going to make over trials. And so we can compare now the dynamics, these amplifying choice predictive dynamics within the activity of the frontal eye fields of our populations of neurons. When we do that during exploitation, we see a couple of things. One, they're aligned to these idealized amplifying dynamics. That's good. This is part of how we think FEF functions, that it amplifies the difference between different things, different choices that you might want to make. The other thing is that if I do it a bunch of times, I see actually really, really similar trajectories every single time. So that's what I've illustrated here as these faded versions of the same trajectories. These are independent samples. When we look during exploration, we see something else, which is that the trajectories are no longer aligned with these idealized amplifying dynamics, and they also look kind of sickly and weird. And I think that that's kind of a powerful idea, that there might be something going on in terms of the dynamics of populations of neurons that hopefully I will have time to get to in a little bit. But before I do that, I want to take advantage of the fact that we are recording simultaneously from populations of neurons to think about what's happening on single trials, right? So because we have this resolution, because we can combine activity across multiple neurons that we're recording at the same time, we can now estimate the extent to which neural activity is in this sort of tightly organized repetitive state versus this disorganized variable state at every moment in time relative to transitions into exploration. So that's what I'm plotting here with, which is what I'm calling the scatter index on the y-axis here. The first thing that we notice is that when you transition into exploration, there's actually a sudden increase in the amount of noise in the system, into how sickly those trajectories look over time. And what's happening here is actually not just like some added noise riding on top of the dynamics of the network. What's happening is actually a disruption of the dynamics of the network. So if we look at, there are typically these long timescale autocorrelations in neural activity. So your brain state at one trial is related to your brain state at other trials. But when those trials cross exploration, when they're separated by a bout of exploration, those long timescale autocorrelations are disrupted. Despite the fact that they're sometimes making the exact same choice between here and here, exploration is basically taking whatever organization was in the network and wiping it out. After exploration, you start to acquire that organization again. You start to ramp back down over time. But it turns out that it's not just a matter of time that causes you to ramp back down. It's actually a function of the number of rewards that you receive. So we can look at that by looking at how the scatter index scales with the number of rewards you've received since you started to explore along the y-axis, or the number of trials. So if it's just a matter of like every time you do something, it ramps down, then the gradient of the scatter index is going to move along the x-axis. But if every reward that you get ratchets you slowly down into this organized state again, that's going to move you along the y-axis here. And that's exactly what we see, that almost all of the sort of variance in this recovery is really driven by each reward you get is ratcheting you down into this organized, repeatable state. So interim summary number 2. Let's see, where am I on time? Okay, I've got some time. So we developed a method to quantify the dynamics of exploration and exploitation that relies on a sort of hidden Markov model, where we model exploration and exploitation as the latent states underlying decision-making. In this view, when you sort of think of behavior this way, exploration becomes a state of its own, driven in part by noise. So its occupancy is driven in part by noise, but also by the rewards that you receive in the world. And it functions to facilitate transitions between different stable policies, to give you that extra moment where you can hop from one state to another state. Our hidden Markov model recovers trial labels that explain a surprising amount of variance in neural activity. And what we do with that is then try to understand how exploration is implemented in the brain. And what we find is that exploration is really disrupting these temporally extended brain states that seem to constrain the variability in neural activity. So, so that was that paper, the 2018 paper. And since then, I've been sort of continuing to dig into this question about what these brain states are actually, how they come about, right? Like what does it mean when I move my hands like this? And what I actually mean is that there are sort of low dimensional constraints on how neural activity evolves over time and across neurons. So in the FEF, neural activity has to move from this place where it's signaling the incoming visual information to this place where it's affecting the motor response, right? And so that mapping, that transition, that path in neural activity from this early undecided point to this point where you've made some commitment is due to a variety of factors, but those factors, you can think of a lot of them as kind of constraints on how neural activity evolves over time. Which is why I put up this image of this water slide here. There are dynamic attractors which cause these low dimensional constraints in neural activity. During exploration, our argument is that those low dimensional constraints are really what's disrupted, and that's what permits the noise that we see in the neural activity, right? So our basin, if you take a slice through this water slide here, we're basically saying that that's sort of flattening out, as opposed to exploitation, where it's really narrow, tight, it's constraining the variability, and ensuring that you precisely execute the behavior that you want to execute at every moment in time. So, so this view makes a couple of predictions for neural activity that we've been sort of digging into in the last few years. One is that neural activity should be more variable. So if the constraints on neural activity are flatter or wider, there's less sort of quenching the variability in neural activity. And the other is that neural activity should be easier to perturb, actually brain and behavior. And that's because if you have sort of a very flat landscape, the same amount of force can actually push you a lot further along than it can in a very sort of tight, constrained landscape. So those are the two things that I'm going to talk about in my sort of remaining time. So in terms of variability, I already showed you that the scatter index is increased during exploration, and so that's one measure of variability. There's also another very classic measure of variability, which is the Fano factor. This is just the variance in the spike count divided by the mean. And one thing we know about the Fano factor is that when you give a network a visual signal, the Fano factor quenches. So the variability is quenched by an incoming visual signal, and then it does some processing, and then it relaxes back up. And so that's what you can see here, is that during exploitation, there's some quenching. During exploration, there's also quenching, but there's a DC offset here, right? So variance is just generally higher during exploration than exploitation. But the other thing that was sort of key for us looking at this was that the amount of time that it takes to recover from that quenching is actually a lot shorter during exploration than it is during exploitation. And I really like this paper from Dean Buonomo's lab, where they actually compare chaotic networks where a low-dimensional attractor dynamic has not been trained into the network to ones where there is a low-dimensional attractor dynamic. And what they see is that in both cases, giving a visual stimulus quenches activity. The chaotic network has higher resting variability than the one with the low-dimensional attractor in it. But the chaotic network also recovers almost immediately from that initial perturbation, right? But provided that perturbation is then causing the network to trace out that repeated dynamic, you're going to have the quenching continue for the duration of time until you get to the end of that attractor. And I think that's what we're looking at here. And we have other measures of variability as well, like the coordination of neural activity of different population trajectories. And this all seems to fit with that broader story. But we can also use computational models to be a little bit more precise about what we mean by the idea of low-dimensional dynamics being disrupted. So in this case, we have a simple competing two-pool attractor network model that's just going to take incoming visual information and make a choice out of it. We can set up the model with strong dynamics that force you to make an option 2 choice and observe the trajectories in the model, or to make option 1 choice. Or we can set up the network with what I'm calling weak dynamics, which don't enforce you to make one choice or another, but just to do something at the end of it. And then we can compare those circumstances with each other, and we can see if that makes predictions for neural activity. There are a couple of things that we notice. The first is that when the forces in the network are strong, when they're forcing neural activity to go to some particular region in space, then trajectories that lead to different outcomes actually deviate really quickly. There's strong forces pulling them apart. The second thing, which I don't know if you can see down here, is the effect of early noise. So if there's strong forces pulling you apart, it doesn't matter where you started, right? You're gonna end up being forced in one direction or another. But if the forces that are forcing you are really weak, and what's happening is you're running into a saddle node, and that's causing you to sort of split off and make a decision at some point. That early noise actually matters. It propagates through over time and leads to very different outcomes. And that's exactly what we see in the data, that there's this crossover where the separation happens more slowly during exploration, but starts earlier, that early noise is propagating through the system during exploration. Okay, so hopefully I've convinced you more or less that neural activity is more variable during exploration, and not more variable in like trivial ways, but more variable in ways that you would expect, given that the constraints on neural activity are smaller during exploration. I'm assuming I'm getting a time check right now. Yeah, I don't know what that face means. Is it like 5 or 2?
You're pretty much out of time, but like I'll let you conclude. Yeah.
Okay, I will conclude then. So brain and behavior should also be easier to perturb. We do a bunch of stuff with this where we actually stimulate neural activity. We find that, so if there are strong forces, our perturbations are actually going to be picked up by the network and routed into like these unexpected off-target outcomes. But if the forces are weak, our perturbations are going to be much more precise and specific, and that's what we find. They're much more precise and specific during exploration. And I think one of the things that we're doing now that I'm actually really excited about is using very, very tiny perturbation currents in the network to actually measure the dynamics of populations of neurons while monkeys are doing these exploit tasks. And I think that's going to be a really powerful way to actually get a better handle on the constraints on neural activity across these different goal states. So I don't have time to talk about any of this. I planned my time poorly. But we did also use naturalistic perturbations of behavior, so just subtly manipulating the visual contrast of the targets. And what we actually found, again, using predictions from computational modeling, was that it seems like what's happening during exploration is not, during random exploration, I should say, during a state where behavior is random, is not that behavior is random. What's happening is that top-down control is actually reduced. So we're specifically removing prefrontal control from the system, but low-level biases that are baked into the system are unchanged, right? So those end up having a bigger impact on behavior when you sort of take out the top-down control. So I think I'm just going to very quickly do a final summary of everything else that I've said. So I started out talking about mice. I said that female mice learn faster than male mice, and that might have something to do with the fact that they're reducing the dimensionality of the world that they're exploring. And then I talked about monkeys, and I said that we were recording from neurons in the monkey brain, and that when monkeys are exploring, when they're using random exploration, we lose the ability to predict what the monkey is going to do from this part of the brain that's classically choice predictive. And I also said that the population trajectories, these sort of neuron dimensional wiggles that seem to support these sensory motor transformations, are disorganized and sickly during exploration compared to exploitation. And finally, that what I think all of this means is that we're basically looking at a prefrontal cortex that's been removed from the computation about what you want to do, right? So insofar as the prefrontal cortex is maintaining this information, this momentary information about your policy, what you're doing is selectively randomizing behavior with respect to specific policies that are learned in the moment. You're not disrupting the knowledge that's maintained in the hippocampus, this map of the world that you have. You're not disrupting these low-level priorities that have been baked into your brain over evolution. You're specifically randomizing behavior with respect to these higher-order policies. So with that, I will conclude and thank. I didn't mention Cindy, who helped with the color and shape rule stuff, or Eddie, I forgot to mention, who helped with the explore exploit stuff. But I think that's all and the various PIs as well. But, yeah.
All right. Thank you very much, Becket. This was amazing. There's a lot of stuff in there.
Yeah.
I'm certainly fascinated by a number of these findings. Can't wait to talk more about this. In the interest of time, let's take one or two quick questions before the break here. First question by Ayuno_N. Do you expect mice to use the same learning strategy or mix of strategies if the stimulus was something that they use more naturally, such as odor, for example?
Oh, that's a really good question. I feel like part of the reason why they ended up, or why it took them, so we were only able to figure out what strategies they were using because it took them so long to learn the task, which meant that we had blocks of 150 trials that we could actually do a lot with. But if they learn the task really quickly, which they might in the case of an odor discrimination, I'm not sure that we would have the resolution to look at these things. But they might still use this early spatial bias strategy. That's an interesting question. Yeah.
Very cool. All right, maybe one more. How do you think brain state neuromodulation affect the energy landscape for exploration? Well, I think you actually kind of addressed that in one of your slides.
Yeah.
So maybe you want to comment a little bit more on that.
Yeah, I think so. I think the most clean thing that I had on there about that was the cocaine stuff. That cocaine seems to deepen these basins that correspond to temporally extended rules. And the other thing is sort of the deepening with every reward that you get, right? The idea that you would sink into these basins in the neural activity. And I think that kind of deepening is probably has something to do with dopamine. Like that would be my guess. And we know that cocaine is actually altering the dopamine system in addition to other neuromodulators like norepinephrine and serotonin. So I think getting into that question of figuring out what the neuromodulatory basis of these brain states are would be really cool. Like, how do we just erase a policy from the prefrontal cortex? Like, that's a big question. And I think neuromodulators are probably involved.
Absolutely fascinating. Okay, so unfortunately, I think I'll stop it here for the questions. Thank you once again, Becket. This is lovely stuff. So I encourage everybody to. So we'll be taking a break now, just before our final keynote lecture of the day by Yves Marder. There is still GatherTown going on. There's a button at the bottom of your screen if you want to go hang out with people. I have a few notes and announcements to make, specifically for GatherTown, and also I would say for Crowdcast. We'd like to encourage people to actually put their real names in the credentials, right? So if you ask questions, say on GatherTown, or GatherTown, if you walk around with your avatar, or on Crowdcast, if you ask a question, it'd be nice to know who it's from. And also in GatherTown, I was asked to let you know that it'd be nice if you left your audio and video on when you bump into people, because that's the sort of the point of the thing. Okay, so that's it. We'll take a quick break. We'll be back at, let's see, 3:30 p.m. For a great the keynote lecture by Dr. Yves Marder. Thanks again.